Good afternoon. Today, I, Dr. Rakesh Kumar, Assistant Professor in the Department of Veterinary Pathology, DGCN Kovas, Palampur, Himachal Pradesh, is presenting in front of you one piece of research work. This research work was basically on one topic, which is effects of root extracts of Saussurea lapa on chemically induced liver damage in rats. And as we are knowing, there are many plant-based treatments which are already known to show better results in order to prevent several ailments globally, and several extensive that searches has already been done in this aspect. And one of the very important plant which is basically grown in the Himalayan areas, and that is Saussurea lapa. This medicinal plant is especially present in certain regions of the Himachal Pradesh, like certain regions of Himachal like Kinnaur, Lahaul Spiti, Chamba, and in India, other than Himachal, it is present in Jammu and Kashmir and Uttarakhand. So this plant Saussurea lapa is one of the endangered that medicinal plant, and this plant is already been known to show several scientific and traditional medicinal properties, and it is used for several ailments, as I told, including cancers. And the medicinal properties of this plant are basically as a result of the constituents which are present in it, and that includes costanoids, dihydrocostus lactone, and cyanopicrine, which are present in the roots of the Saussurea lapa. And there are several studies I told, like this plant was used as an antioxidant agent in several studies against tumors, and even cardioprotective effect of this plant has already been known. And in certain hepatoprotective activities, this plant has already been studied in several experiments in several scientists. And in my study, we had on the basis of the previous literature, we had planned this study with two objectives. One was to study the protective effect of Saussurea lapa on certain biochemical changes in the chemical induced hepatic damage, and second is to study the protective effect of Saussurea lapa on pathological changes in the rats which were induced with one chemical which is DEN. And what we have done, we had collected this plant, the roots of this plant Saussurea lapa, from Lahaul and Spiti, and the identification of this plant was done in CSIR IHPT institution at Palampur. And after the collection of this plant, we had shade dried this plant, we had prepared 70% equithanolic extract, and after that we had done the lyophilization, and finally the characterization was done by UPLC for the presence of the bioactive constituents which are present in this plant. And in a brief methodology, we had also covered the in vivo study, and in this in vivo study we had taken albino vistar rats, and these albino vistar rats were induced with DEN, and we had given 3 different doses of the plant extract which I had already mentioned, that is Saussurea lapa, and the animals in this experiment were recorded for the clinical symptoms, and several other protocols like imaging technique like ultrasonography to assess the liver damage in the rats was done, and finally the sacrifice was done, and we had collected blood samples and liver tissues for the further investigations on in vivo experimental study. And this is the brief methodology. This is the Saussurea lapa plant we had collected from Lahaul and Spiti. These are the roots. This is the fine ground powder of root extract, then extraction 70% equithanolic that extract was prepared. This is the extract, and this is the protocol by which that extraction was done. And this is the in vivo that experimental protocol we had followed. We had taken 46 that male albino rats, and we had divided these rats into 6 groups. The first group was given with the control, and it was provided with that vehicle. That vehicle was basically a solvent, DMSO, and it was given for the to the rats through oral route for 10 weeks. The second group, it was induced with one of that liver damaging agent, and that is DEN, and this was also given through oral route. Third group was given with the liver damaging agent DEN, and along with this we had given silymarin, which is a hepatoprotective effect, already known hepatoprotective effect. And this comparison of these this plant extract in 3 different doses was done with silymarin mainly. And the fourth group was given with the first dose of this plant extract after producing liver damage through DEN, and this plant extract was given at the dose of 100 mg per kg through oral route. And second, the dose was 250 mg per kg, and third dose was the highest dose of this plant extract, and that was 500 mg per kg body weight. And this experiment was basically conducted for 10 weeks. And I told that UPLC, characterization was done to identify the presence of the bioactive compounds which are present in the plant roots mainly. And these that constituents which were present in this plant root was costanoid, which were present in the highest amount, and followed by the dihydrocostus lactone. And these are the chromatograms we can see. This is the standard chromatogram, and this is the chromatogram which is showing the peaks of these two compounds. And we had conducted the acute toxicity study as well, where we can see we are not getting any alteration in the liver tissue on gross examination and even on the histological examination for the 2 weeks. And we were providing the dose of Saussurea lapa at 2 gram per kg body weight, and we are not getting any pathological alterations. And after that ultrasonographic examination, that is one of the imaging technique in the animals, it was done, and we can see in the imaging techniques we are getting marked lesions in the group which was provided with the den only, and we can see this demarcated hyperechoic area in the liver parenchyma, and this is very large area which is involved basically. And if you're doing the comparison of the den induced group with the silymarin or the highest dose of the extract, we are getting promising results. And gross pathology, we can see on gross pathology, this is the normal control group where we are not going to get any lesions, whereas in den induced group we are getting a large liver with nodular hobnail like that appearance we are going to get. And if we compare this den induced group with the highest dose and the other groups treated with the Saussurea lapa, we are getting a dose dependent response or reduction of the lesions. And these are the lesion scores. We can see the group treated with the highest dose of the extract, it showed minimal gross lesions if you are going to compare with the den induced group or silymarin induced group. And liver weight, we can see the liver weight was significantly increased in the group provided with the den, and it was reduced in the treatment groups in a dose dependent manner, that is group SA, SB and SC. Hematological examinations, when blood examination blood profile was done, we had seen that values of the hemoglobin, PCB, TEC and these WBCs count, they were decreased in the group provided with the den, and it was reduced in the groups provided with the root extract, and these alterations were minimal. And next, these are some of the graphs which are showing the hematological alterations. And biochemical results, we can see that values of the ALT, ALP were significantly increased in the groups provided with the den and decreased in the group provided with the highest dose of the extract. These are some of the pictorials or graphical manifestations we can see. And these are the histological changes that the normal control group. And here we can see the den induced group. It shows histological changes to an advanced extent, and this includes pseudolobulation, fibrous tissue proliferation and bile duct hyperplasia. And here we can see similarly the bile duct hyperplasia is evident in this group, binucleation, and these increased areas they were evident. And silymarin group, in silymarin group, these changes they are going to be reduced, and these were significantly reduced in the groups provided with the extract, and these alterations were minimal in the group provided with the highest dose of the extract. And these are the lesion scores. We can see the maximum lesion was present in the group provided with the den. And after that we had done mesenteric staining, and this blue color we can see this is indicating fibrous tissue proliferation, and it was maximal in the group provided with the den only. Immunological expression of caspase-3 was significantly evident in the group provided with the den, and it was minimal in the group provided with the highest dose of the extract. And finally, from the study, we had concluded that the highest dose of the root extract of Saussurea lapa has hepatoprotection, and it is proposed to be better than silymarin against den-induced liver damage. And biochemical parameters and other lesions, histological lesions, they are indicating the foundation of Saussurea lapa as a wonderful hepatoprotective agent, and creates an ample scope for its exploration in the human and veterinary medicine. And this is the graphical representation. We can see this den, when the den has given to the rats, we are getting the metabolites of this product. It leads to ROS production, reactive oxygen species, lead to apoptosis, and alteration in the liver enzymes, that gross changes, microscopic changes, and leads to liver damage finally. But the Saussurea lapa has reduced all these gross production, apoptotic changes, microscopic and gross changes, and it will lead to finally to reduction in the liver damage. Thank you.